Professor Andrew Owen from Liverpool, and he will talk about application of nanotechnology for optimization of antiretroviral drug delivery.
Thanks, Juliana. And thanks again to Alejandro, who's going to make my life a lot easier by having already covered many of the key areas. So I'm going to echo a lot of what has already been said. Just by means of an overview, I'm going to talk a bit about formation of solid drug nanoparticles to overcome drug delivery challenges for drugs with poor water solubility. Talk quite a bit about our development program in Liverpool for antiretroviral solid drug nanoparticles. Look a little bit about the opportunities for passive and active targeting in HIV, and then hopefully I'll have time to go into some of the early validation that we've done with hyperbranched polydendrons, which are a new class of material. So grinding pharmaceuticals has been around for a number of years. It is what it says on the tin. It's the physical fracturing of large fragments of drug to generate small fragments of drug. It's typically done by using a mill. This is a picture of a Zeta mill, and works by putting large fragments of drug into the mill, applying a lot of energy to grind, and grind again until you end up with very small fragments of drugs which are measurable within the nano range. It's known as an attrition technique. It does have some limitations. It's limited to materials with specific properties. For example, it's limited to crystalline APIs, and there's a requirement for a high melting point because of the heat generated in the mill, which can cause melting of low melting point APIs. There's limited approved excipients for the approach. In some cases, there have been reports of impurities from the milling agents, or in some cases from the mill itself. And there's clearly a correlation between the amount of energy placed into the system and the size of the nanoparticles which are generated. But it has proven successful for a large number of drugs. I think the last time I counted, there were around 28 FDA-approved solid drug nanoparticle formulations on the market. It's shown benefits across a number of different disease areas. For example, in the case of Ritalin, combination of this milling technology with other technologies enabled transformation from a twice-daily regimen into a once-daily regimen, which mirrored a twice-daily regimen. For morphine, again, you've got this sustained release of oral administration of solid drug nanoparticles. In a number of cases, it's been shown to overcome food effects, so the PK being acceptable in the fasted state here for the nanoformulation, but not for the conventional formulation. And for other agents, it's been shown to improve the bioavailability and therefore increase the amount of drug which is able to enter the systemic circulation after oral dosing. So solid drug nanoparticles have proven to be successful, but there are limitations with the milling approach for their production. So the approach that we've been using in Liverpool is termed emulsion-templated freeze drying or spray drying. I'm not going to dwell on this because the method is published, but it's a bottom-up approach. So unlike milling, it forms nanoparticles from the bottom up rather than from the top down. You form an emulsion of an organic solvent with an aqueous solvent. The organic solvent dissolves the drug. Subsequently, when you spray dry or freeze dry, as the liquid comes out of the emulsion, you're left with a dry, porous structure with zones of the organic material or the drug within that structure. And when you subsequently add water or an aqueous phase back to that porous structure, you release the drug as solid drug nanoparticles which are in suspension. So physically, the solid drug nanoparticles that we produce using this approach resemble those which are produced by milling. So these are some preclinical formulations of an antiretroviral ripivirine. These are two antiretrovirals, efavirenz and Lopinavir solid drug nanoparticles, and you can see that we're within the same size range here as those formulations produced by a pharmaceutical company. And using scanning and electron microscopy, they're not dissimilar in structure when you visualize them either. There are some advantages to the approach. I'll just run through a few. For example, we can use crystalline or amorphous API, whereas milling, you need a crystal in order to mill. Because we're not milling, the melting point is irrelevant. We've got a wide access to approved excipients. Using a freeze-drying approach, we can cover a lot of ground very quickly, so we were able to make lots of different options from very small amounts of material, whereas using a spray-drying approach, we were able to rapidly scale to kilogram scale. We've got a one-step formation of a solid format, whereas milling produces a liquid format. And getting back to the point about cost, we estimate that for our efavirenz lead formulation, this is going to cost between $4 and $16 per kilogram, which effectively translates to around $4 per patient per year to the cost of an antiretroviral formulation. So just to recap on a few of these things, the two drugs we've chosen are efavirenz and Lopinavir. Efavirenz is currently globally first-line therapy. Lopinavir is a second line. Again, there's over 34 million people infected with HIV globally, mainly within resource-limited settings such as sub-Saharan Africa. And importantly, for these drugs, current global manufacturing capability is being outstripped by patient need. So we simply cannot make enough of these drugs to meet demand at the moment. There's a real need for new formulations for children. In some cases, Lopinavir, for example, is given to very young children in a 40% ethanol solution, so effectively giving a child their drugs with a shot of vodka. And so clearly any strategy that reduces doses required for effective viral suppression will increase the number of patients able to receive therapy and may also reduce costs. Patents on both these drugs, I think, have actually expired now, so that opens up opportunities. But there is a caveat that there are preferred regimens emerging in resource-rich environments. So to run through the approach, we use binary combinations of a polymer and a surfactant, and we can generate a large library of solid drug nanoparticles in a couple of days to a week's time. Characterizing them, they range in size, mainly submicrons, so between 100 and 500 nanometers, with a range of surface charge from very positively charged nanoparticles down to very negatively charged nanoparticles. And one of the first pharmacological analyses we did was to look at the transcellular permeation across a CACO2 cell monolayer, which models gut epithelium, so looking at the gut compartment and movement of the drug to the blood compartment. And one of the first things that we noted is when we looked at each one of these different solid drug nanoparticle formulations, there was a relationship between the size of the nanoparticles and the ability of the drug to permeate across the CACO2 cell monolayer. There were also some relationships with certain excipients used, but I don't have time to go into that. The other things that we did was model this in vitro data using physiologically based pharmacokinetic modeling. The PBPK base model was validated against existing clinical data for efavirenz, but then we were able to plug in the in vitro data, and we showed that in the simulation, the best formulation would be predicted to have this effect on the PK, whereas the worst formulation could actually make the situation worse. So we do need to understand the drivers of the benefit in this type of approach. Our PBPK, the guy leading our PBK modeling is Marco Saccardi, and he's going to give a comprehensive talk on this tomorrow. So we then moved with our lead formulations to scale the drug loading up to 70% drug relative to excipient, and then conduct a series of rodent studies relative to the standard preclinical formulation of efavirenz. And you can see that similar to the PBPK model, we saw an increase in the pharmacokinetic exposure of efavirenz delivered as a solid drug nanoparticle compared to the conventional formulation, with very little difference in the tissue-to-blood ratio across different tissues. We then go back to our PBPK modeling, and we were able to show that a predicted dose reduction of 50% would match the C trough value for efavirenz, which is the most vulnerable part of an antiretroviral regimen, PK profile. So we've also done a whole set of work on Lopinavir, which I'm not going to go into in detail, but in this case we were able to make solid drug nanoparticles containing both drugs, so both Lopinavir and ritonavir. The PK profiles were pretty equal across the two drugs, although there might have been different, less variability for the nanoformulation. But these may have implications for pediatric dosing. This isn't Lopinavir, this is a dye, but you can see that when you add the dye to water, in the normal form, you just get a messy precipitate. Whereas when you, I'm not sure whether that next video gonna run. Yeah. So when you add the processed dye, so this is an insoluble dye, when you add the processed dye to water, in this case you can already start to see it forming a dispersion on the surface of the water. And when the fingers come in to give it a mix, you can see that you can form a nanosuspension of this insoluble material in water, which has got to be favorable to a 40% shot of ethanol for pediatric dosing. So we've now scaled and translated using a Nyromobile minor spray dryer to kilogram scale. We've confirmed the drug loadings for the final product and the physical characteristics, which are stable. The drugs are amorphous, confirmed by XRPD, and we've had filled capsules and bulk powder on GMP stability and storage now for around 3 months. And we're planning to start a healthy adult volunteer bioequivalence study to attempt to establish bioequivalence from a lower dose later in this year. So there are some opportunities in HIV as well for some passive and targeting approaches. For example, macrophages are a huge site of virus replication, and certainly at some size ranges they're phagocytose particles. There's also some data to indicate that HIV-infected macrophages exhibit a much higher phagocytic activity than uninfected macrophages. So there may be an opportunity there for a passive targeting into a major site of HIV replication. We all know that there are potential benefits for getting into lymph nodes, which are another important site for HIV replication. But there's a range of different active targeting moieties available as well. We've got a number of cell type specific proteins we could think about targeting with aptamers or antibodies such as CD4 or CCR5, for example, and tissue-specific receptors as well. Similar to other diseases, there's interest in getting into the brain, for example. The testes is another important site. So we've done some studies of the solid drug nanoparticles. We've shown that particularly in immune cells, we seem to get a much higher accumulation of efavirenz when incubated as a solid drug nanoparticle compared to a solution in monocytes, either transformed cells or primary monocytes, also into CD4, CD56 cells, as well as into macrophages. We tend to get a higher accumulation. We've shown this using fret-based dyes to show that in macrophages, it's intact nanoparticles which become internalized. And even over a PK profile for efavirenz, so over a 24-hour period, the amount of efavirenz within the macrophage is still almost twice as high for the solid drug nanoparticles as it is for when incubated with a dissolved molecule. And in cell-free based systems, so the ability to inhibit reverse transcriptase is identical between the solution and the nanoparticle. But in the cell-based systems, we see the shift in the IC50 indicating a higher potency for the solid drug nanoparticles, which correlates with the degree of enhanced intracellular accumulation in the cell types used in those assays. So that's our solid drug nanoparticle platform. Just going to spend a few minutes going over some of the recent work that we've been developing, a new platform technology called hyperbranched polydendrons. I'm not going to go through this in detail, but Dr. Lee Tatham has got a poster here at Clinam on board 59 if anybody wants to take a look at it. But the strategy here is to use a dendron initiator with a monomer and a brancher to form a polymeric nanoparticle in a cheap and scalable way, which hopefully retains some of the surface functionality of a dendrimer. These are very new materials. First patents are just being granted on the hyperbranched polydendrons. So this is quite an interesting topic. If we take a good solvent such as acetone and combine that good solvent with the polymeric nanoparticles with a bad solvent such as hexane, you can see that at low solvent fractions of hexane, we get this shrinking in size as the nanoparticles reach colloidal stability. However, when we hit about 0.2, we get this massive jump in the size of the nanoparticles, which is consistent with a controlled aggregation into a larger nanoparticle, which then subsequently again condenses in size up to around 0.8 solvent fraction of hexane, followed by further aggregation thereafter. And you can see that at the low hexane concentration, we've got a very broad PDI, which subsequently narrows when we get into this range here of around 150 to 200 nanometers in diameter. We've also got some additional flexibility. This year is conducting the nanoprecipitation by adding THF into water to form an aqueous nanodispersion. We've also incorporated dendron and PEG initiators to form polydendrons with dendrons and PEG at the surface of the particle. And this data just summarizes this. And just to say, by modifying the initial and final concentrations of the materials, as well as the ratio of the dendron to the PEG, we can get to a size range between around 80 nanometers to 200 nanometers without losing control of the polydispersity, which is important. We've also done some physical characterization in terms of the SEM for these materials as well. We've probed the internal architecture of the nanoparticles. So by using Nile red incorporated into the nanoparticles, Nile red changing in fluorescence in response to its lipophilicity of its environment. So you're getting a higher fluorescent here, but also using pyrene, which reports on the polarity of the environment of the pyrene as well. You can see that down here, using the I1, I3 ratio, we're able to tune the environment of the nanoparticle to reach polarities of its environment somewhere between the chloromethane and water by changing the G2 dendron to PEG initiator. When we move these into CACO2 cell assays, so again looking at the apical to basolateral and basolateral to apical transport, we see a correlation between the G2 dendron molar fraction and the permeation across the CACO2 cell monolayer. We can control other things about the polydendrons. We can use different generation dendrons, for example. We've done this with different size peg chains from 750 to 2,000 grams per mole. We can change the degree of polymerization for the internal polymer, the architecture. We've made pH-responsive polymers in the internal core as well. And we can also add some functionality to the dendron. For example, we can add sugar groups or amine functionalized dendrons. If we look at the degree of polymerization on the internal architecture, by increasing this, we see an increasing the permeation across CACO2 cells. It's decreased by the amine functionality, the movement across the CACO2 cell monolayer. And this kind of relates to the degree of accumulation within the CACO2 cell. So the amine functionalized polydendrons have a lower accumulation within CACO2 cells, but interestingly, they have a much higher, much better accumulation into macrophage type cells, in this case, the ATHP or activated THP1 cells. So just to summarize, I think in diseases like HIV, there's a real need for alternatives to intravenous delivery because intravenously delivered drugs have already failed in HIV due to the need for an ongoing commitment to daily dosing for the rest of the patient's life. Our EFTD represents an alternative approach to formation of solid drug nanoparticles, which may have advantages to traditional approaches. Our first generation SDNs now have the stability data to underpin our regulatory submission, and we're hoping to move these into bioequivalence studies later this year. And our early work, I think, certainly warrants further evaluation for the hyperbranched polydendrons. I'd like to thank all the people that have funded us for this research. Marco Siccardi for the PBPK modeling, Neil Liptrot, who's conducted a whole set of immunological analysis on these materials, which I didn't have time to go into, but he does have a poster here on poster board 73. Lee Tatham, who's done all the pharmacological evaluation on the hyperbranched polydendrons. Paul and Melinda from the Clinton Health Access Initiative, who've given us an enormous amount of support with the regulatory aspects, and Scott and Marina at NCL, who've given a lot of help to Neil in the immunological evaluations. Thank you.
Thank you very much, Andrew, and also Alessandro. Do you think that the cost when you use this formulation could decrease in HIV therapy?
So if we validate the 50% reduction in dose while maintaining bioequivalence, currently efavirenz, generic efavirenz costs around $52 per patient per year. So if we reduce those drug costs down to $26 by half in the dose, the additional processing is predicted to add around $2. So it's not a 50% reduction in cost, but it's not far off.
But I am wondering whether the manufacturing cost and the price has any relationship. Do you know that? I just don't know.
The majority of the costs are due to the synthesis of the API. But you're right, there are additional costs which aren't factored into that. And we haven't done a robust cost effectiveness analysis yet.
Thank you very much. Alessandro?
Thanks for your nice presentation. I just want to add a comment to your question. And probably now we are talking about drugs that are already in the market for almost 20 years. But if we transfer successfully in any of these technologies to drug that is newer and we are able to reduce the dose, we are not talking about $25 a year per patient. We are talking probably about several thousands. So the idea, I guess, for you too is to be able to implement a nanotechnology platform in HIV and then to transfer, with some modifications, a platform to other newer antiretrovirals.
Agreed. And the other thing to note on that is we're not talking about one drug for a patient. We're talking about at least 3 drugs for a patient. So that has to be factored in.
Yeah, my question about the packaging capacity. The dendrimer usually have that kind of limitation. For yours, only one layer, not like Tamela's multiple layer dendrimer. So capacity is the issue or not the issue? And do you have the polymer drug ratio limitation?
Yeah, so and the data, I didn't show this data, but for Lopinavir, we now have 20% drug-loaded hyperbranch polydendrons for that material. So for the hyperbranch polydendrons, we're working at around 10 to 20% drug loading.
No more questions? Thank you very much, Andrew. And I would like to thank for every speaker and the audience for attendance. So coffee break.